Good morning everyone. My name is Rodern F. Pedlawan from BS Ed 3. Our topic for today is all about the process of mitosis and meiosis. And what is mitosis? Mitosis is a process of nuclear division in eukaryotic cells that occur when a parent cell divides to product to identical daughter cells. During cell division, mitosis refers specifically to the separation of the duplicated genetic material carried in the nucleus. And we have a parts or stage of mitosis. First, we have a Prophase. Prophase is the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus known as chromatin condenses. And we have a pictures of prophase. And second is we have Prometaphase. Is the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. During Prophase, the physical barrier that encloses the nucleus called the nuclei envelope breakdown. And we have a Metaphase is staged during process of cell division. Individual chromosomes cannot be observed in the cell nucleus. However, during metaphase of mitosis or meiosis, the chromosomes contains and become distinguishable as they align in the center of the dividing cells. And we have Anaphase, the process that separates the duplicated genetic material carried in the nucleus of parent cell into two identical daughter cells. During Anaphase, each pair of chromosomes separated into identical independent chromosomes. And we have in Telophase. During Telophase, the effects of Prophase and Prometaphase are reversed. And we have a meiosis. And what is meiosis? It's a process where a single divided to twice to produce our cells containing half the original amount of genetic information. These cells are our six cells sperm in males and of course eggs in female. And now we have and meiosis can be divided into a stage. These are divided between the first time the cell divided meiosis 1 and the second time it's divided in meiosis 2. And number 1 is we have interphase 1. What is interphase 1 in meiosis? The DNA in the cell is copied resulting into identical full set of chromosomes. Outside of the nucleus are two centrosomes, each containing a pair of centricles. These instructions are critical for the process of cell division. And we have a picture of interphase of meiosis here is centrosome with centricles, aster, nucleus, chromosome, nuclear membrane, cell membrane, and centricles. And of course, we have Prophase 1. The copied chromosomes contains in two X shapes, insertion that can be easily seen under a microscope. Each chromosome is composed of two sister chromatids containing identical genetic information. The pair of chromosomes may then exchange bit of DNA in a process called recombination or crossing over. And we have Metaphase 1. The chromosomes pair line up next to each other along the center or equator of the cell. Centricles are now at opposite poles of the cell with the meiotic spindles extending from them. The meiotic spindle fibrates attack one chromosome of each pair. And Anaphase 1. In each meiosis 1 is the sister chromatid stay together. This is different to what happened in mitosis and meiosis 2. And we have a picture. This is separation of daughter chromosomes and sister chromatids attached to the centromeres. And we have a telophase 1 and cytokinesis. The chromosomes complete their more to opposite folds of the cell. At each pole of the cell, a full set of chromosomes gather together. And we have a Prophase 2 and meiosis. In each of the two daughter cells, the chromosomes contains again into visible X shape instructions that can be easily seen under a microscope. And Anaphase 2, the sister chromatids are then fold to opposite folds due to the action of the meiotic spindle. These separated chromatids are now individual chromosomes. And Metaphase 2, in each of the two daughter cells, the chromosome pair of sister chromatid line up into each along the equator of the cell. Meiotic spindle fibres at each pole of the cell attached to end of the daughter cells. Telophase 2 and the centrokinesis, the chromosomes complete their move to the opposite pole of the cell. At each pole of the cell, of a set of the chromosomes gather together. And that's all for today. Thank you.